All right, everybody. Today we're going to talk about DNA, and we've kind of already talked about the structure of DNA. So we're actually getting into genetics and heredity. So basically, how DNA codes for your traits, and how they can be passed on and stuff like that. So we got to do some basic DNA stuff first to kind of remind you and get a basis for what we're going to learn in genetics. So there we go. I got a bunch of different animals I'm going to put up on the screen here for you, or just a bunch of pictures of living things. I want you to think about what they have in common. We've got a platypus, possibly the weirdest animal there is. Snake, shark, dinosaur, zebra. Look at those stripes. And then that's bacteria. Okay, so hopefully you're already on to me because you're like, well, you just said we're going to talk about DNA. So yeah, like that is what they all have in common. But I want you to think about how cool that is, that these things all have DNA in common. Even these dinosaurs that are all dead, R.I.P. But they all of their traits, as different as their traits are, right? Like the platypus has a bill. Like that is weird. The scales of the snake, the body structure of the snake. Sharks have like a weird cartilage skeleton. Dinosaurs, they have all kinds of different traits. So anyway, and actually, sorry, I'll mention bacteria too, because bacteria are so different than any of these other things. All of these other things are like brothers compared to the bacteria, which is like a weird cousin or something, okay, or even less related than your cousin. But anyway, they all have the same 4 letters of DNA that we're going to kind of talk about today. And you probably learned before the A's, T's, G's, and C's. Those 4 letters are the key ingredients to DNA that if we put them in different orders and have different lengths of our chromosomes of those 4 letters of DNA, then that gets us all of these very, very different organisms. And it's the same 4 letters. And I think that's pretty interesting. Okay. And when we talk about evolution, we'll kind of get into that a little bit more. Okay. But anyway, so that question was, how do the animals get their traits? DNA. And so before we actually knew that DNA was a genetic material, people had to do experiments to try to figure it out. And so this is kind of an interesting one that I wanted to show you. And let's go through it real quick. So 1928, we didn't actually know that DNA was genetic material. And so this is one of the ones that helped get us there. So in experiment 1 here, we have what we call our R cells. This is just a vial of bacteria. And the bacteria, we are going to take it and we're going to inject it into a bunch of mice, and we see that they live. That's nice. And then experiment 2, what we're going to do is take this different strain of bacteria. It's related, but it's a different strain. We inject it into the mice, and take a guess, they die. Okay. So the next thing that we can do is we take this bacteria right here, the green one, that we already have established is dangerous. I don't know what's happening to my arrow. That we've already established is dangerous, and we can kill them by just heating them up. And so you heat them up, the cells of the bacteria all kind of break apart and they die. Okay. And we know this to be a viable method to kill bacteria. And we take this once it's cooled down and we inject it into the mice. And the mice should, what's our prediction? Okay, they live. Like they should live. We killed all the bacteria. Okay, so we've done these 3 experiments, and this kind of helps us set up for this 4th experiment. We have these conditions that we know of already. So we know that these R cells here are safe. What is happening? We know that the R cells are safe. We know that the S cells are deadly. We know that the S cells that are killed are safe. And so now what we're going to do is we're going to take some safe bacteria. We're going to take some deadly but killed bacteria. Both of these conditions we know are safe for the mouse, but when we mix them together, so what we're going to do is kill these, and then they break apart all of their cell contents, including their DNA, is hint, is spread out into there, and then we mix this with this guy. And based on what we already know from previous experiments, this mouse should live, right? We are taking two different things that are safe for the mouse and giving them to the mouse. However, what happens is that the mice die, okay? And or according to this picture, it looks like they're kind of just sleeping, but they die. So what's happening here is what the researchers found was that there is some chemical that when we break apart these deadly cells that is getting into the safe cells, the non-deadly cells, and is giving them the trait of being deadly. Okay, and so what we kind of later figured out is that there is DNA that like breaks apart in or the cells break apart, the DNA is floating around in here, and the non-deadly bacteria, what they can do is they take it into their cells and then they use it to gain a new trait. Okay, this process is actually called transformation because they are transforming themselves to have a new trait. It's like we as humans can't do this, right? It would be like if there's some DNA lying around on the ground that gives you the trait of being able to fly, and I was like, oh, I want that, and so you pick it up off the ground and you inject it into your body, and then it's like, bam, you can fly. Okay, there's actually like some video games you can probably imagine. Like in video games, you can do this kind of thing. You like inject some DNA into your body, and then you gain a new ability for the video game, right? That's what's happening with these. Or that's what happened with these bacteria that became deadly. They gained the deadly They gained the deadly DNA from those other bacteria. So this helped researchers discover that there's some chemical that is the genetic material, and it helped them understand that bacteria can even take it up from their environment. And this kind of laid some of the groundwork for later on when we discovered what the genetic material was. Okay, so make sure you have a little bit about this experiment written down here. Okay. Here's a takeaway. There's a chemical that transformed the non-deadly strain into a deadly one, and that is DNA. That's not exactly what they got from it. They didn't know it was DNA yet, but we know now that it is DNA. Okay. All right, structure of DNA. We've done this before, so I want to go over this quickly. So nucleic acid, we've gone over the 4 categories of macromolecules, but Na of DNA is nucleic acid. Okay, you should know that. Our examples are DNA and RNA, which we're going to talk about more in the coming weeks. And the monomer, so the individual piece, remember DNA is a long-stranded molecule, so the individual pieces of that long strand are nucleotides, and they look like this. They have 3 parts, this, and this. But I like this picture better because it reminds us that they're not just random shapes. There actually are different atoms. So we have oxygen, phosphorus, hydrogen, nitrogen, carbon, and did I miss one? No. Okay. So we have a bunch of different atoms that make up these nucleotide molecules. So on this next screen, what I want to show you is a little bit more close up of this double helix. So we have a red strand and we have a blue strand. Okay, so that would be the red strand. Oh, that's a blue, and the red strand there. Okay, and we also have these parts in the middle. Okay, that meet up. So we have to account for that when we're looking at the different nucleotides here. I want you to know the different parts of a single nucleotide and how they make up this structure. So the strands, we got this and this part of the nucleotide, make up the rails of the ladder. And sorry, just a sec. Okay, and this part here, this is the base. So this is one nucleotide that I've just outlined, but the green part is the half of what makes up the rungs here. Okay, so this is one nucleotide, and it always pairs up with another nucleotide on the other side, and together these, the green parts, make up the rungs, and then this, the phosphate and the sugar, oh, this is going to be hard. There we go. Phosphate and sugar make up the rails. Okay. So in total, I want you to try to see how many nucleotides there are in this picture and write them on your notes. So how many nucleotides? I want to see that written. So I've just outlined 2. How many more are there? Okay. All right. So, ignore that for a second. Okay. So this guy named Chargaff, he was studying DNA, and he was looking at different organisms, and he would look at their DNA, and he was trying to figure out the amount of different kinds of DNA, right, of the different letters. And so what he found was that there's always the same percentage of adenine and thymine, so A and T, and there's always the same amount of G and C. So in any organism that he looked at, there was always the same amount of A and T, the same amount of G and C. And so, my question is, what do these results tell us? Okay, and if you can't really understand what's going on in this first sentence, I put a little analogy here. So it says, if you studied all of the shoes in the world and found that there are always the same amount of left shoes as there are right shoes, what would you conclude about shoes? Hopefully, hopefully you figure out that, oh, well, maybe shoes come in pairs. Maybe left shoes and right shoes go together. That's why there's always the same amount of left and right shoes, provided you're not losing one shoe all the time. So anyway, what it's going to tell us, so we go back to Chargaff, what that's going to tell us is that A's and T's, since they always the same amount of them, they come in pairs, and G's and C's also come in pairs, right? And so I have a question for you here. First, I'm going to show you this picture. So if we have a green one, there's always a red one. If there's a yellow one, there's always a purple one. There's a key there for you there. Right? So hopefully that kind of makes sense. Now we can do this question. It says, if there is 20% adenine in an organism's cells, if you break open the cells and you count the percentage of adenine, the A molecule, what percentage of cytosine is there? Okay, so hold on, under Chargaff's rule here, you can put this pairing rule, right? So A is going to pair with T, and G is going to pair with C, and that's also what we see here. So this is an A, and the red is a T. And so this was a C, and this is a G. So now we can do this question. If there is 20% A, and all you need is a little knowledge of percentages, what percentage of T should there be? Well, there should also be 20% thymine, okay? And together, this is going to make up 40%. And when we're dealing with percentages, obviously we have to go to 100. So we have 60 left, 60%. And 40% is 100. So if we split that into 2, we have 30 and 30. And so we have 30% G, 30% C. Okay, so our answer is 30% cytosine. Okay, I'm going to ask you questions like that, so make sure you get that example down. Cool. Let's keep going. Okay, now what we have here, I just want to remind you of what a chromosome actually is, because we're going to kind of look at some chromosomes. So a chromosome is made of DNA. So if we take this DNA here and what we're going to do is it wraps around these proteins here, which are called histones. It wraps around those, and then those wrap around even farther, and then it coils and coils and more. It's actually called supercoiling. We finally get a chromosome structure. So this entire chromosome is made of densely coiled DNA that is just like packed together very, very tightly. Okay, so just want to remind you of that structure because that's going to help us here. So chromosomes have genes. So here is a chromosome. And whenever we look at these, sometimes they have these bands on them. And so what we are kind of showing, or what those pictures are kind of showing, is that chromosomes have different genes. So this is a segment of DNA. So let's say this is a segment of DNA, and it is a gene which codes for a a protein, and maybe like one down here, this band, let's say, is the second gene. Okay, and to zoom in even further, we have what we are known as base pairs. So just like what we showed before, a C always pairs with a G, and an A always pairs with a T. So this is, if we zoom in on the DNA, it's full of these base pairs. Okay, and remember, each one of these is a nucleotide. So that's the G nucleotide, and that's the C nucleotide, and they pair together. Okay. Cool. Yeah, I like that picture because it just breaks it down. And so base pairs. So here we have, let's see, 1, 2, 3, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15 base pairs, which in the scheme of things you're going to see is not very much. Okay. Here are some examples of traits, but we will not go over traits right now. Just suffice it to say that each gene can help to give us a trait. All right. This is kind of cool. This is an image that shows a chromosome. Actually, this is chromosome number 9. And it says, the first thing to note here is this says 136 million base pairs. So in the image before I showed you, we just counted 15 base pairs. Well, chromosome 9, which is not even the largest chromosome, has 136 million pairs, which is incredible. And here what we see is a bunch of different diseases that are associated with the genes found on this chromosome. Okay, so there are a lot of different genes found on this chromosome, and when there were mutations in these genes, we can get some of these different diseases that are listed here next to this chromosome. So I just want to show you this picture of human chromosomes. So we said that was chromosome 9, and they're listed in order of length. So chromosome 1 is actually the largest. So chromosome 1 looks to be about double the size of chromosome 9. So we can imagine there might be 200 million base pairs on chromosome 1, which is a lot. Okay, and so 46 chromosomes total, 23 pairs. I also want to show you some chromosomes from other animals. And so, like, this is a dog, and it says this has 78 chromosomes, right? So 39 pairs. And this is a fruit fly, which has 8 chromosomes or 4 pairs. And so I want to emphasize that chromosome number doesn't really have anything to do with complexity or anything. It's just the way that the DNA is arranged and broken up. Okay, just thought that was kind of interesting. All right, last thing here. Okay, so here's what you're going to do. Last thing in your notes, I want you to draw this chromosome here. Okay, so draw like a stick-like thing with a bunch of bands, because that's how we represent a chromosome sometimes. And then draw this little arrow, and then you have a double-stranded, a double helix. And I drew it in 2 different colors to show our 2 different strands. And so here I drew one strand of the DNA in blue, and I want you to do 2 different colors or 2 different shades or something. So blue, and then I want you to draw the complementary, the base pairs here. So if this is a C, this is a G. This is a G, that's a C. If that's a T, that's an A. What's the last one I need to do? That's an A, that's a T. Okay, so go through, you got to do all of them. I skipped some, and do all of these. Okay, on this side, what I want you to do is, it says list 10 traits that you have genes for. You can look some up if you need to. Don't use these. I want you to come up with your own 10 traits that you have genes for, that like your genes code for in you. Okay, so you're going to list them on this side, and you can kind of like be like, just pretend like, oh, that gene or this trait goes with that band right there, because that is the band. That's a gene, and it codes for that trait. Okay, so I want you to make this kind of diagram in your notes. All right, guys, thank you very much. Let me know if you have any questions. Bye.